Welcome to Signals and Synapses, the podcast of the Max Planck Institute for Biological Cybernetics in Tübingen, Germany. I'm Sophia Jans, and today I'm joined by Jennifer Lee, Drew Robson, and Svenja Klinkowski. Drew and Jen have run a lab together for over a decade, currently here at the MPI for Biological Cybernetics. Welcome to this episode, Jen and Drew.
Thank you.
Nice to be here.
Svenja is a PhD student in neuroscience at our MPI and at the University of Tübingen. Svenja will be co-hosting this episode and talk to Jen and Drew about why zebrafish are so important for neuroscience, what we know about how they think, and how this compares to us humans. Thank you for joining us, Svenja.
Thanks for inviting me.
Many of our listeners have probably already seen zebrafish in private fish tanks or aquariums. But the little striped fish also become increasingly popular with researchers. Why is that?
Well, I would say that the zebrafish community started with development because it was possible to see from a single cell all the way up to a complicated multi-cell organism with organs and even a brain. And then finally, because of that last point, the neuroscience community also became interested because you have single-cell access to every single neuron in the brain.
Yeah, it actually started in Tübingen. So Yanni, Lucy, and Walhart started using zebrafish as a genetic organism for studying development because of the access to all of its cells.
Great, that sounds very interesting. So what are the specific research questions that you want to answer with your zebrafish?
So I think like Sophia said, we've been running a lab together for a decade, but it actually goes back further than that. So when we were grad students, first year of grad school, actually, we were looking around at neuroscience, and it seemed like the big problem with neuroscience is that the brain is too complicated. There's too many cells. They have very complicated connections with each other, especially if you start thinking about things like how do you store internal representations of the world? How do you plan trajectories when you're navigating? These are kind of computations that require what are called recurrent networks. So networks where the cells do not just simply feed forward to another set of cells and so on, but they actually feed back on themselves. And so the brain is full of these, recurrent loops. And the loops make it very, very hard to dissect. And the problem is kind of exponentially harder the larger the brain is and the less access we have. And so when we started as grad students looking around, it just seemed like there was no organism where you could see the activity of all the cells. And so instead, what you were getting, for example, if you look at a rodent brain, you have about 100 million cells, but you were accessing at the time a few hundred at a time at most. And that was like the most advanced technologies that we had. And so if you were coming into this and just saying, what is the ideal system you would want to have? You would want a system where you could see the activity of all of the cells and maybe even map out all of its connections. And the only vertebrate animal where that's possible is zebrafish. And that's still kind of true today. So a larval zebrafish has about 100,000 cells. All of it is optically accessible. So using a microscope, you can see the activity of all of the cells at the same time. And now we can actually even start to map out the connections between the cells. And so it just seemed like from kind of like a first principles reasoning kind of place, it seemed like the right system to start with if we were to understand how the brain works.
Can you give us a little idea for those of our listeners who are not very familiar with the human brain, how much smaller that is actually?
Oh my God. So a human brain is 100 billion. And so, it's many, many orders of magnitude bigger. And so I think that, with neuroscience, you need all of these systems because they all have their own unique advantages. So for humans, of course, there are things that, like language. There are abilities that's really somewhat unique to humans. That's very hard to study in any other system. And so even though we don't have very good access to a human brain, you still want to, study humans. But it's the whether, I don't know when we will get to a point where you can actually say, I'm recording from every cell in the human brain. I'm not sure that will happen in my lifetime. I think I definitely believe it will happen one day, but I think it's not anytime soon.
Yeah. Cool. So if I have understood this correctly, you can basically investigate any relevant behavior that's also interesting for neuroscience in general, just in the zebrafish, and only the behaviors, I guess, that you can observe in zebrafish.
Yeah, exactly. So that's the really good point. The really good point is that, like I said, humans, like we can do so much. It's really fascinating when you study human behavior. And then when you go down to smaller and smaller animals, and it's a really interesting evolutionary question, it's a really interesting computational question, is that when you start to work with really small brains, really small computational systems, like a little zebrafish brain, what can they do? That's where you kind of have to figure that out, right? So you go, oh, here's a system that's perfect because we have all of the tools to see the entire circuit, but what does this circuit actually do? And that took us many, many years to figure out. I think that's not just us, but our entire community. And that is probably still very mysterious, I think, to a lot of people, because, with psychology in humans, that's something that's been happening for a few hundred years. I would say even farther back if you count like philosophers. And then for rodents, people have been doing, we've been running rodents through mazes since, like the Toman maze in the 40s. So it's been like 100 years of research into exactly what are the kinds of behaviors they can generate, the complexity of the decisions that they can make, and even that is still an open field. Zebrafish, started as a developmental organism, and it's only in the last, I would say 20 years that people have really started to look at, what can this very tiny larval zebrafish actually do. And so when we were grad students, we had to first, show, they're capable of something like operant learning for the first time.
Absolutely. So there are, of course, as Jen mentioned, certain behaviors and capabilities cognitively that only humans can model. And of course, those are amazing. But if you think about the multifaceted problem of life, of being alive in a complex, rich, dynamic world, every animal has to solve these problems at some level. And of course, when animals have such different scale and complexity, the solutions may not be identical, but at some level, the animals have to solve these problems. And so we try to put animals that we study, the zebrafish, into naturalistic settings and simply see how they solve the particular problems that they have to solve. And so we find that they are progressively better throughout development, throughout their life, at solving the multifaceted problems that they face in the world. This starts from early things like learning spatial representations and learning which parts of the world are good or bad. And then later we see social capabilities emerge. And then in adults, maybe even some more complicated cognition. And so we'd like to access all aspects of that.
So that means you are investigating the different stages also. So you investigate larvae as well as adult zebrafish?
Not so much adults right now. So we have a collaborator who we're actually working closely on adults. But for spatial and social cognition, we're mostly looking at from larval to juvenile stages. And so until late juveniles, zebrafish is still completely transparent, which means that you can still see all of its cells. At some point from juvenile to adulthood, then they grow, bone, skin, they become very big, and that makes it much harder to access the entire brain. And so I would say that you still have whole brain access into juvenile stages. And that's when we actually see the most rapid emergence of, for example, social cognition emerges very rapidly from the first sort of 3 to 4 weeks of life. And we also see learning rates actually increase dramatically over that exact same stretch of time. So it's a really, so we are looking across development as these kind of cognitive abilities come online, but I don't think we're looking all the way into adulthood right now.
Maybe you can explain a little how you're actually observing the larval zebrafish. I mean, they're transparent, all right, but how do you actually observe what's going on in the brain?
Yeah, there is a sensor that essentially detects neural activity in neurons. And this is a sensor that can be genetically expressed. So basically, we make a transgenic animal that expresses the sensor in every cell in the brain. And then the sensor gives off light when that cell is active. And we can, if you put this animal then under a microscope that can see all of the cells, then just by seeing which cells light up, you can trace out which cells are active active when the animal is thinking or making decisions or, navigating.
Yeah, cool. Okay. And maybe to understand this better, how does it work? Like, in application, can you maybe go into the details of your recent paper that you have published in Nature?
So this builds on our tracking microscope technology because we finally had the ability to record the neurons, as Jen has just described, comprehensively across the brain. But importantly, we could do this under the conditions when the fish was simply swimming around in the world. And so with the benefit of hindsight, to be honest, we should have been asking the question about spatial representation as soon as we got this microscope working. But nobody, including us, actually thought that these animals represented space in the explicit way like we see in mammals, these things called place cells, where a cell is active at only one particular location in the environment. But because we had the technology, of course, eventually we got curious and we just had to check. And to our great surprise, we actually found that there was this exquisite population of cells, and particularly in the telencephalon, which is like the forebrain, the front of the brain. And so that seems to be a very key center for the fish, even the larval stage.
Yeah, pretty cool finding. Can you maybe explain what you mean by place cells? What does it mean?
Well, actually, there's a very nice mathematical way to understand this, because since the early 90s, people were able to say, what does it mean for a cell to quote unquote know something about where the animal is in space? And so there's an equation, it doesn't matter, but the idea is that you can derive simply from first principles, how much information would you get if you knew that a certain cell had just been active at a specific instant in time. And so just from first principles mathematical derivation, you can actually put a number on how much information you would get from knowing that that cell was active at an instant in time.
So if I try to break it down, would a place cell, let's say in us humans, be like a cell that fires when I'm standing in front of the Eiffel Tower, and another place cell fires when I'm standing in front of a church that I know?
That's right. So there will be different cells depending on where you're standing. But in fact, there are additional things that we want to know about the cell before we're willing to call it a place cell. So for example, if you are looking at the Eiffel Tower, but you only get the activation of this particular cell when you're looking at it from a certain point of view, we regard that differently than if that cell was active, in fact, whenever you're standing at that spot where you were looking at the Eiffel Tower. But regardless of whether you're looking toward the Eiffel Tower or in the opposite direction away from the Eiffel Tower. And so, in fact, one of the hallmarks of a place cell is that it doesn't care so much about which direction you're looking. What it really seems to care about is where you're standing in space.
You have mentioned already that place cells have already been shown in other species, in mammals, in humans, rodents. What makes it so special that you now found them in fish?
Yeah, that's a really good question. So it's been found in the amniotes, so birds and mammals. And so there's two reasons why it's great to find them in fish. One is just the evolutionary question, right? So there is, place cells are thought, not just, I think Drew's answer is good, that they are a representation of space, but they are also considered to be kind of like the unit of memory. So episodic memory is thought to be encoded also in the hippocampus, and place cells are thought to really bind together, your memory of the environment with the knowledge of where you are in space. And so it's computationally very important, and it's not known when the evolution came up with a neural circuit that can actually generate place cells. And so if it's really true that they are unique to mammals, you would say that there's some architectural change in the brain that happened when we evolved, during the branch point between, reptiles and mammals that uniquely gave it the ability to generate place cells. If it was happened far earlier, like we're seeing in zebrafish, it would suggest the possibility, at least. This could be convergent evolution, but it would suggest the possibility that the neural circuit that support place cells emerged actually much, much earlier in evolution. And it actually evolved a much smaller nervous system as well. And so that there is some kind of a minimal circuit that can generate place cells. So going back again to the idea of brain scale, so this is kind of my second point, is that a mammalian brain, a bird brain, a human brain, these the hippocampus in these animals are enormous. So you need millions of cells potentially to, so you have millions of cells and the place cells are embedded within this enormous circuit. In fish, the structure where we find these place cells have only about 10,000 cells. So it really is like I really think most likely the smallest possible computational network that can probably generate place cell like properties. And because it's so small, it means that you can really see the entire circuit at the same time. And you have very uniquely now tools then in zebrafish that you don't have in other animals, where you can actually map out all of the connections between the place cells, between each other, and also between the place cells, their upstream inputs and their downstream inputs. And that gives you an enormous amount of power to solve this circuit in a mechanistic way that you really cannot do right now in any other species.
You mentioned the telencephalon in zebrafish being like the relevant structure, and you've also mentioned the hippocampus in humans, also in rodents. Is it like the analog in a zebrafish, or what does the telencephalon encompass?
So it's a good question. We don't know for sure. So we know that the telencephalon is likely to be analogous to the precursor to the mammalian hippocampus and cortex. But it is very hard to say right now, and it may be fundamentally, I think, conceptually impossible to say, is the hippocampus is the zebrafish telencephalon the hippocampus or the cortex? I think that's unlikely. I think it's likely to be something that really is just older than any of those structures, but it can carry some of the functions that are now carried in the hippocampus and cortex.
Super interesting. So the human hippocampus, also in rodents, for example, is thought to be the key structure, not only for spatial navigation, but especially for memory. And you have also mentioned that maybe cortical structures or cortical functions are also represented or located in the telencephalon. And cortex or cortical functions are so, like, there's a lot of functions to the cortex in humans, especially like different kinds of cognition, also memory, and self processing, all of these kind of human functions of the cortex. And I was wondering, do you also investigate things like memory in zebrafish, and where on a neural level would these functions be located?
That's something we're looking at right now, actually. So in terms of the behavior, we started looking at memory when over 10 years ago. And what we've known for a long time that they can form memories of space. So if you punish them in a particular area, they will avoid that area. If you reward them in a particular area, they have a tendency to go back to that area. And so that is something that we know they can do. We are now actually currently doing experiments where we have place preference assays that we've set up where they have to find, for example, a safe region in space. This is not quite like the Morris water maze. It's a little bit simpler, but that's the basic idea. And we're looking at the neural signals that continue to, for example, encode the safe zone, even when the stimulus itself has disappeared, right? So that the animal can't just be using their sensory feedback to know where they should go. And we actually... we already actually know that not only are there cells in the telencephalon that are active at the regions where they've been punished or at the boundary between the regions where they've been punished and not punished, but that when the animal then goes into like a quiescent state where they're not moving around, they can actually reactivate those cells that seem to be associated with the areas that the place cells that's associated with the areas that they're punished, which would actually really do suggest that they have representation of what's good and bad in the environment, and that is actually a memory that they can potentially retain.
So when you say quiescent state, when they don't move, does that translate to what would we call sleep in other animals like humans?
By the definition that currently exists in zebrafish, which is that they stop moving for a minute, and this is the formal definition of sleep currently in our field, then they are asleep. I think that's, again, one of those things where why an animal stays still for an extended periods of time could have many different functions. And so does it have the exact same functions as humans? I don't know. I mean, it's possible if these cells are being reactivated during this quiescent period, that this is actually something that will be useful for memory consolidation, for example. And that's something that we know to be true in mammals, but that's something that we would need to show in zebrafish. And there are very cool experiments you can do. For example, you can actually go into these quiescent states when the animals are not moving but replaying events from the past. You can perturb the activity of those cells and then see if the memory disappears when they wake up or is degraded when they wake up. And so those are kind of future experiments that we really fun to do. But until that happens, I think we can't say for sure right now.
So the paradigm that you have just described to investigate memory, for me, it sounds a bit like that there's always some kind of emotional component involved. If you use like a paradigm where you punish the animals, is that something that also is reflected in the neural activity?
So that's a really good question. As I was answering your question, I was thinking to myself, yes, that's one way we know that to show that, they have a memory because they can form a place preference. But in fact, if we look at the neural data, you don't need that emotional component because of the fact that, they make a map when you introduce them into a new environment, and it's a neutral environment. There's nothing good, nothing bad. We're not explicitly punishing them or rewarding them. And they make a map. And when you take them out and put them into a new environment, they make a new map. But you can then return them back to the old environment, and you can see that they recover the map they had initially made, which already suggests that even without a very emotionally salient event, they just, perceptually, they are just constantly building up representations of their world. And I think that's true for humans too, right? We're constantly, our memory is not something that's formed because, we had a nice snack and that's when memory is formed. We're forming memories constantly of the world that we're in. Some events might be more salient, and so it changes what goes into long-term storage or not, but we are actually constantly forming these kind of memories of where we are. And also and I guess the other thing that we see, though this maybe is a little early days still, is that they also seem to have memories of landmarks and objects. And so things that are associated with, not with exactly where they are in space, but for example, with an object in space. And this is just an object. It's not a bad object or good object. It's just a interesting looking object in the world. I mean, having said that, though, maybe the deeper answer to your question, though, which I don't think that anyone in the field has a good answer for, is that while we think of, punishment and reward, explicit punishment and reward, so giving food, giving shocks, as very emotionally salient, you could also argue that innately a lot of things are emotionally salient, right? But just not, it's not something that the experimenter is giving, but that as an animal traverses the world, uncertainty could be anxiety provoking itself. And that's not, and so when you go into a new environment, you could say that that's always emotionally salient. And if something changes, that could induce, again, stress because of uncertainty or the violation of prediction. And so there's actually a really interesting, there's an interesting direction in neuroscience right now of like, what is sort of innately just, there are these neuromodulatory signals that's associated with kind of like reward punishment with emotional responses, for example. But these signals tend to be actually maybe are always active to some extent.
Yeah, for sure. I agree. So even a freely behaving animal would always encounter like locations where they find food, for example, which would be rewarding, and also where they encounter a dangerous situation, which would maybe be like punishment or a situation where they would be anxious. And that maybe brings us to my next question. In which way would the situations that you investigate be the same like from a freely behaving animal?
So that's a great question. And thinking about that question and your previous question, I think one of the interesting things about thinking about naturalistic behavior is that it's not necessarily the case that everything in the world always has to be rewarding or punishing. There can be information that might just be good to store away because it might come useful later. So we think about this, especially now that we have the ability to record at all the cells in the brain at the same time that we also record the behavior of the animals. So whereas in the past you might have been motivated as an experimentalist to put in rewards and punishments because then you get an overt behavioral response that we can actually see, even if you did not have recordings of the brain. Now we have the recording of the brain all the time at the same time that we see the behavior. And so then you can ask the question in a different way. And I think this is what Jen was talking about also before, that she wants to set up situations where the animal can just explore the world, learn things about the world that could be useful later. And one example of this that I could give you that we're thinking about a lot is learning safe locations in the environment so that later on, if the animal is threatened, perhaps it remembers there was this spot that I could run away to that would be safe. And at the moment that it sees this safe location, initially, it may not have any particular value in the sense that we didn't try to provide a reward or anything. But because there's something that in nature, the animal has this propensity to just map out locations and know something about them based on maybe the experience of the ancestors of this animal. And so as a consequence of these kind of strategies that allowed the species to propagate and this animal that's in front of us today to even be here, because of this, we can now ask, do these animals actually store this kind of information for future use? And so safety learning would be a really cool application that we're exploring now.
Yeah, I think that's true. I think safety learning, it really extends across all species. I mean, and how they pick a safe location. So there's a really interesting study from Nahimowski's lab who studies bats. And so he released bats onto a desert island, and which is just an insane experiment. It's amazing. And what he finds, this was a conversation with him, so it's not published, but what he finds is that they always go back to one spot. And it doesn't seem like there's anything special about that spot, but they keep coming back to it. So it's not, so exploration is never random in animals. And this also made me think of studying, I think, rodents back in the 90s, which was also just a behavior only experiment where they showed that if you put a mouse, if you take a rat and you put it into a new environment, wherever you drop that rat, they will just keep coming back to it, even though there's no reward, there's no punishment in the environment. But it's almost as if the position where they've placed this animal becomes, like a safe spot that they keep kind of navigating back to. And so the navigation itself, even though it's, you could say it's just random exploration, isn't random. They're always coming back to a kind of like a spot they've designated as, I guess, a safe zone. And that's something that we also see in zebrafish, is something people have seen even in flies. And so I think those are questions that really extends across all the species.
Yeah, that sounds like a super interesting approach for sure. Are there any other challenges in your research? What's like the most difficult part of investigating zebrafish behavior?
I'll take the technical part, and then maybe Jen can talk about from a more behavior or neuroscience perspective. But technically, the truth is that we always want to go faster and longer, and we want to see just every cell in the brain at the highest recording rate that we can, but also really to push the duration of time that we can get these recordings. And since we've really hitched our wagon, so to speak, to this use of fluorescent indicators in order to read out the neural activity, the single most important technical thing that we need to overcome is what is called photobleaching, which is simply that when you shine light on these fluorescent indicators, they get dimmer over time just because you kind of wear them out. And so it would be extremely helpful for a lot of the scientific questions that we're trying to go after to simply be able to extend our observation time to maybe an entire day or even multiple days of development.
So Drew wants to do you know the movie Truman Show?
Yeah.
So Drew wants to do that for zebrafish. So, from when it's born all the way for the rest of its life. Maybe not. So there is actually a related species to zebrafish called Danionella cerebrum. And it's kind of like zebrafish, but it never grows up. And so even in its adulthood, it's fully transparent. It's the size of a juvenile fish. So you could imagine, with something like that, you could really do the Truman Show. So you follow it from the time when this animal is a single cell all the way into adulthood, all the way into old age, but the only way to do that is if you have sensors that don't just become dimmer and dimmer and eventually disappear over time. And so but yeah, if you can get over that technical problem, you could actually do it.
Yeah, so the truth is that we are making great progress in the lab, mostly on the microscopy side. On the sensor side, we wait for great breakthroughs from the field, but in the meantime, we can do as much as we can just by making the microscopes more efficient. And in fact, we can get orders of magnitude more performance in that way. So that's our current path forward.
Okay, so now after talking about challenges, maybe you want to pinpoint the advantages again of investigating your behaviors in zebrafish, maybe just like a quick summary. Why is the zebrafish such an amazing model organism for neuroscience?
I think it's the Goldilocks principle. The brain is just the right size. We really need to have the access to all the cells and also get the wiring diagram so we know exactly how these cells are connected to each other. And that's just a starting point to have any hope of understanding all these recurrent loops that Jen was talking about before. And so at the scale of the zebrafish brain, we can do this. And then with the foundations we learn, perhaps we can inspire people to eventually do the same when the tools are ready in mammals and beyond.
And that brings us to the end of today's episode of Signals and Synapses. A big thank you to Jennifer Lee and Drew Robson for sharing their insights and expertise with us, and to Svenja Klinkowski for co-hosting and contributing her insightful perspective. It's been such a pleasure having you on the show. And of course, thanks to all of you, our listeners, for tuning in. As always, you can find links with further information in the show notes. If you enjoyed today's conversation, be sure to subscribe and share this episode with your friends. Until next time, stay curious and keep listening.